authentic, you know. So, you are 100% sure that entire India helicarpa is there, but it is also having variation about 1%, right. So, selection criteria should be kept based on the projects. Organization of samples, it up to you how you want to organize. पैसा ज़्यादा है तो multiple plates you can use. If you have less money, individual specimen you can sequence, that's up to you. Of course, the next steps are really crucial where we don't do it in practical. Okay, I just want to highlight this sample digitization. How many of you are pathologists here? Okay, very good. So, fungal identification कैसा होता है भाई? Sequence बोल मत बोला प्लीज. माइक्रोस्कोपिक आइडेंटिफिकेशन होता है फंगस. माइक्रोस्कोप में क्या देखोगे? स्पोर्स देखोगे और भी देखोगे ना, ओनली स्पोर्स से थोड़ी आइडेंटिफाई हो जाएगा. माइसीलियम ग्रोथ होगा. माइसीलिया में क्या देखोगे? सेप्टेशन देखोगे. ये सब फोटोग्राफी तो करना पड़ेगा ना, दैट्स वेरी इंपॉर्टेंट. सब इंसेक्ट पूछूंगा तो इंसेक्ट तो करना ही है हमें, बट at the level of fungus you have to do photography. Okay. So, sample digitization is very important. I told you we have collected 20 samples of insects, coded. Each sample has to be digitized. 1 to 20 I am giving coding, no, 20 specimen. All 20 we have to digitize at the level of specimen. Okay. So, that is the first, and of course we have done labeling, imaging those specimens. Next, tissue selection and labeling tissue samples. So, I am just skipping. So, next wet lab. Now, you have done this wet lab. Okay. So, tissue homogenization ho gaya, lysis overnight, DNA extraction, it changes from a sample what you are taking. Okay. PCR, primers different for different groups, Sanger sequencing. Now, even it is not only Sanger sequencing, now even you can also use NGS for DNA barcoding. Okay. So, short reads भी लेके बाद में उसको contact बना सकता है, that's not a big deal. Nowadays, if you are whichever is cheaper, you can go with that. Okay, so यहाँ से हम अभी इस class start करेंगे. So, it's already 10 minutes. So, I will just give a brief and I will show you. I have already have 1 or 2 small projects in bold. I will directly log into the bold and we will see once you get these sequences from the company. Okay. So, what happens in India, sending sequence is cheaper than doing yourself. Okay. So, 550 rupees में आपको एक one side का sequence मिल रहा है. So, why you have to spend time on this? Okay, you have to process that properly and send for sequencing. अभी क्या किया आपने, PCR हो गया, चेक कर लिया इलेक्ट्रोफोरेसिस में? नो, चलो आ ही गया 650 बेस पर आपका सीक्वेंस आ ही गया, इलेक्ट्रोफोरेसिस में आपके चेक भी कर लिया. कैसे भेजोगे सीक्वेंसिंग के लिए? अभी बोला नहीं ना किसी ने. PCR हो गया, अभी 500 माइक्रोलीटर का PCR रख दिया आपने. कितना रखा? अभी कितना रखा? 10 माइक्रोलीटर का 25. यार आपने रिएक्शन वॉल्यूम कितना था टोटल? 25. 2 बैच सेपरेट है क्या? ओके ओके सॉरी. ओके फाइन. सो 50 ले लो. ओके. सो इलेक्ट्रोफोरेसिस के लिए आपने 3 माइक्रोलीटर या 5 यूज किया. चलो 5 यूज कर लेंगे. सो 45 बच गया आपके पास. सो 45 माइक्रोलीटर में 45 बेचने का जरूरत नहीं है. 20 माइक्रोलीटर इज एनफ. अनदर 20 माइक्रोलीटर यू कीप विथ यू. सीक्वेंस फेल्यूर होगा तो और एक बार बेच सकते हो. मेनी टाइम्स व्हाट स्टूडेंट्स विल डू, 45 कर दिया हमने, 45 भेज दिया, सर सीक्वेंस रिपोर्ट नहीं आया. नेक्स्ट टाइम वंस अगेन यू स्टार्ट डूइंग PCR. When you are doing PCR, be careful or do it for 50 microliter and around 15 to 20 microliter is enough for Sanger sequencing. Okay? Sanger sequencing आप PCR भेजते ही PCR तो नहीं करेगा, वो sequencing तो नहीं करेगा, उसको प्यूरिफाई भी करना पड़ेगा Sanger sequencing के लिए. ठीक है? So purification kit मिलता है, PCR product purification के लिए kit मिलता है. If you want to use kit and do it, that's fine. Otherwise, pay another 150 rupees extra. Company people will also do that purification part. But you should be aware of that. there is another part called purification, because आप PCR में क्यों purification important है? PCR में क्या क्या डालते हैं? Components तो पता है ना, so इतना component भी इंडर करेगा ना sequencing में, so purification important है, ठीक है? So इट्स आल्सो हेल्प्स इन एनरिचिंग, आपका जो 650 बेस पर जो DNA एम्पलीफाई हुआ है उसी को एनरिच करेगा, सो रिजल्ट्स क्वालिटी कम नहीं होगा. ओके फाइन. सो सीक्वेंस हो गया अभी. सो व्हाट, हाउ दे विल सेंड सीक्वेंस? डायरेक्ट फास्टा फाइल में भेज देगा. एक एक कंपनी एक एक तरह भेजता है. एक कंपनी आपको ईमेल में जिप फोल्डर भेज देता है, एक कंपनी एक लिंक भेजता है, आपको जाके पोर्टल में रजिस्टर करके आपके अकाउंट में डाउनलोड करना पड़ेगा. इट डिपेंड ऑन कंपनीज. देयर आर थाउजेंड्स ऑफ कंपनीज नाउ. ओके, सो मोस्ट ऑफ द टाइम यू विल गेट रॉ फाइल्स. फास्टा एक उसमें साथ भी रहेगा. ओके, रॉ फाइल्स रहेगा, फास्टा भी रहेगा. और रॉ फाइल आने से डरना नहीं है. देयर आर सो मेनी टूल्स, यू कैन जनरेट द फास्टा फाइल. ओके, इट्स वेरी इजी. सो वंस यू गेट द सीक्वेंसेस, द नेक्स्ट थिंग इज हमारे पास 2 मेगा डाटाबेसेस है. जो वन कॉमनली यूज्ड मेगा डाटाबेस कौन सा है? जेनबैंक, NCBI, नॉट जेनबैंक, NCBI. ओके, सो जेनबैंक इज अ पार्ट ऑफ इट फॉर जस्ट सबमिशन. ओके, सो द इजीएस्ट वे टू चेक एनी बारकोड और एनी सीक्वेंस, उसका क्वालिटी होगा, वो सीक्वेंस ठीक है नहीं है, इज जस्ट ब्लास्ट इन जेनबैंक. ओके, सो उसमें बिकॉज इट्स अ ह्यूज डाटाबेस, you imagine that it's like a big ocean. Okay, and bold is just अभी एक छोटा river है. only insects का, कुछ small genes का एक group बना के रखा है. Okay, but our aim is not, it's very difficult to go and search anything in an ocean. It's somewhat easier in a river. So bold is more easier and more accurate because उसका data कम है and focused on barcodes. Okay, that's why we are concentrating on bold. So first you can just check in GenBank and sequence quality check कर सकते हैं. I think अनिर्बन सर का क्लास है होगा ना. So अनिर्बन सर will take a wonderful class. आपको 2.5 hour he will really explain very well how to handle the sequences, basic analysis कैसा करना है. I will not explain anything about that because that class is really useful for you. He is a wonderful teacher. Okay, so uploading sequence data and image specimen. There is a lot of difference between uploading a sequence in bold and NCBI. First you should be sure about it. अभी तक जो first 2 स्टेप हमने बड़ा बड़ा लंबा स्टेप्स देखा ना, उसमें जो इंफॉर्मेशन जनरेट किया आपने वो दोनों में यूज होगा. बट इन GenBank they don't ask you for any images, anything, only sequence data and what is the, who identified the specimen, latitude, longitude, location. That is enough for GenBank submission. That's why all our students, those who don't want to work more, they will submit to GenBank. Okay, and it's easy. For bold you should have very well curated sequences, very well documented data for your sequences. So you have to prepare in Excel sheet all those data for uploading. Okay, and the good thing with bold is it auto validates your sequences. आपका raw file upload कर सकते हो along with FASTA. The quality of your sequence will be auto validated. That's a very good thing with respect to bold. And if there are any stop codes, कोई भी sequence में stop codons का बहुत problem होता है. It's very common, especially cytochrome oxidase 1. When you are using universal primers for barcoding, it's very common that pseudogenes amplify हो सकता है. Okay, so those, there are so many bioinformatics tools to identify. Okay, so that's a entirely different class. I am not going into that, but you should be aware of that there will be some problems for sequences, but it can be fixed. Okay, so once you upload the data, so what you have to do? Analysis. So एक sequence कर दिया, उसका identity आपको हेलिकॉर पर पता चलने से you are not done with that, no. Okay, how you can utilize that sequence to analyze what is the nearest species of Helicorpa, what is the difference between the neighbor of Helicorpa and Helicoverpa from Delhi. Okay, those analysis you can do. That's a BOLD has so many analysis tools integrated in that system. That's the beauty with barcode of life database. And they will allot a bin number, barcode index number they will designate. So, to explain in a simple words, bin is nothing but if you are submitting like 500 sequences of Helicoverpa armigera, all 500 sequences they will put group into a one section. They will give number to that one. For example, a Helicoverpa armigera from India and New World like America and all, they have more than 2-3% difference. They will still name it as Helicoverpa armigera, but they will give 2 bin numbers to it. That means if there are 2 bin numbers, that means bin 1 and bin 2 are different from some like diversity, like 3% margin or something. Okay? So, it varies with groups. In entomology, we use around 5%. In some groups, they use only 3%. Okay? So, these are bins. So, it is a group of sequences. They will give bin number, and also the good thing with BOLD is whatever sequences you have uploaded in BOLD, in a click you can submit to NCBI also. Okay? So, now both are integrated. So, easily you can submit, because you are submitting so much of data to BOLD and it is auto validating your sequences. And some institutes they want NCBI sequences accession number. So, they have made this option that you can submit to NCBI. चलो अब हो गया, you imagine that you have submitted, आपका project हो गया. ऐसा नहीं है project हो गया घर चले गए. So, you have to once again go back and post processing things you have to do. Why these are important? It's for data validation. Whatever data you have generated, next if somebody is coming and checking that data, it should be available. Okay, what is the purpose of research? Once you do research, you publish, somebody should repeat that one. ऐसा नहीं आपने कर दिया, आपका लैब में ही काम कर रहा है दूसरा लैब में काम ही नहीं कर रहा है वो प्रोटोकॉल. Okay, so post processing involves cataloging of remaining tissue. I told you we have to catalog DNA samples, storage of tissue and DNA samples in DNA libraries, maintaining inventory of sequence data. Nowadays what we will do is, if one of the specimen, if I barcode it, I will whatever the code I will give from the beginning to end, I will just attach that code to the specimen itself along with label. That is the easiest way. Okay? So, if anybody want to check that BOLD may, if you type that specimen code also, you can retrieve that data from BOLD. That is the easiest way to do it. So, it is a very quick way of summarizing. 15 minute लगा इसको summarize करने में. Okay. So, now I just show you think I have logged in. Yeah. क्या हो गया भाई इसको? अभी तो था, मैं लॉगिन नहीं वो आपने जो करा निकाल दिया क्या? हमारा बंद हो गया. ठीक है, डायरेक्ट कर दिया था मैं, आपका अभी ज़रूरत नहीं है बोल के. Any of you are working on DNA barcoding? No. ITS में भी कोई नहीं कर रहा है क्या? पैथोलॉजी में भी कोई नहीं कर रहा है? सॉरी? ओके ओके. पॉपुलेशन जेनेटिक्स में भी कोई काम नहीं कर रहा है क्या? किसमें काम कर रहे हो आप लोग? ओनली मैनेजमेंट और जीनोमिक्स? हाँ? हाउ मेनी ऑफ यू आर वर्किंग ऑन मॉलिक्यूलर एस्पेक्ट्स? सब लोग काम कर रहे हैं मॉलिक्यूलर में. क्या काम कर रहे हैं? रैंडमली बोलो क्या काम कर रहे हैं? मॉलिक्यूलर जेनेटिक्स भी नहीं कर रहे, ये भी नहीं कर रहे, जीनोमिक्स, ट्रांसक्रिप्टोमिक्स, यार ट्रांसक्रिप्टोमिक कर रहे हो उसमें मॉलिक्यूलर जेनेटिक्स नहीं है. हाउ डू यू आइडेंटिफाई फंक्शंस? ट्रीज, ट्रांसक्रिप्टोम से भी ट्री बनता है, पता है? रिलेशनशिप समझ सकते हो? बस काम नहीं कर रहे हो बोल रहे हो. मैं वही बोल रहा हूँ, बार कोड एक जीन करने से बार कोडिंग नहीं है भाई. नाउअडेज पीपल आर डूइंग जीनोमिक्स फॉर बार कोडिंग. होल जीनोम दे आर डूइंग. बार कोड इज जस्ट ए fingerprint for a species. It may be a one gene or it may be a entire genome. But what you are using from that? Okay, the idea is, see, it's a genomic is just a tool. मैं बायोइन्फॉर्मेटिक्स को साइंटिस्ट नहीं मानता हूँ. मेरा सबसे बड़ा दोस्त बायोइन्फॉर्मेटिक्स से है, ठीक है. बायोइन्फॉर्मेटिक्स जस्ट ए टूल, वो कोई भी यूज कर सकता है. एंड आज nowadays bioinformatics has become so cheap, you can easily generate lot of data, but after that what you are going to do is important. What question you are going to ask. Okay, so question क्या address कर रहे हैं वो important है. So tools which are you are using is okay, but whole purpose क्या है, that is important. So that's why I don't differentiate yourself for transcriptomics or genomics. Okay, the question you are asking is important. हाँ, कोई पूछा नहीं मैम.
कोई नहीं पूछा. So already you have done the ICAR and you will do the molecular identification. Then what is the difference between DNA barcoding and molecular identification? Now this is question for you and you and between solve. And you give answer, otherwise why you are not taking this question? Whether it's two different things or the same thing, like in the different name or any difference is there. Don't think about the database. Database is a database.
ठीक है भाई, अभी मैम ने क्वेश्चन पूछ लिया है, मैं आंसर नहीं बोलूँगा, आप ही बताओ. सी डीएनए बारकोडिंग कब आया? 2003 में आया. क्यों आया? पॉल हर्बर्ट ने एक प्रोजेक्ट लिखा, उसको प्रोजेक्ट करना था, आ गया, है ना. So molecular identification was there from 1800, like 1899 something. आइसो एंजाइम्स यूज करते थे. जब से डीएनए भी छोड़ो, प्रोटीन जब एक्सट्रैक्ट करना स्टार्ट किया ना एंजाइम्स, तब से मॉलिक्यूलर आइडेंटिफिकेशन है. व्हाट इज द एडवांटेज वी आर गेटिंग हियर ओवर दैट? आपको क्या एडवांटेज लगा? हाउ मेनी ऑफ यू एक्सट्रैक्टेड प्रोटीन्स? सर किसी ने ठीक है वेस्टर्न प्लॉटिंग किया है, किसी ने एलएसए तो किए होंगे पैथोलॉजी में. कितना टाइम लगेगा भाई? अप्रोक्सीमेटली कितना टाइम लगेगा? वेस्टर्न प्लॉटिंग के लिए कितना टाइम लगेगा? ठीक है, दैट इज वेरी वेरी शॉर्ट. शी इज सेइंग वेस्टर्न ब्लॉटिंग एक एक दिन 2-2 दिन लगता था, अभी सब किट आ गया है, जल्दी हो रहा है. ठीक है, द पर्पस इज दिस इज सो क्विक एंड यूनिवर्सली इट कैन बी अडॉप्टेड इन एनी लैब. एनी लैब. अभी पॉल हर्बर्ट कनाडा में ही हैज डन इट, शशांक यहाँ पे आपको सिखा रहा है ना. सो मोस्ट ऑफ द टूल्स दे केम इन बिटवीन, दे आर नॉट रिपीटेबल मोस्टली, ओके, and they were not adoptable in most of the labs. One is this repeatability and adoptability and cheaper, very very cheap. Like 1000 is nothing in molecular biology. 1000 rupees you are getting forward and reverse sequence, okay, and kit के लिए extra ले लो 20,000 का 20, अभी 10,000 में आप 50 रिएक्शन कर सकते हो. So calculate कर लो, okay. So 50 सैंपल्स वहाँ पे आपने सेव कर दिया. सो अराउंड 1800 में even including your electrophoresis, you're identifying one species at molecular level. Is that cheap? Okay. But still my answer didn't address madam's question. ये तो cheap हो गया, पॉपुलर हो गया, डीएनए बारकोडिंग, अडॉप्टेबिलिटी इजी हो गया, इसीलिए सब लोग बोलने शुरू कर दिए इट्स ए बारकोडिंग. सो काफी है टैक्सोनॉमी में. ठीक है, वो भी कंसिडर कर लेंगे. ये टर्म यूज नहीं करते हैं, मॉलिक्यूलर होलोटाइप. अच्छा टर्म है, वी कैन अडॉप्ट इट. हाँ, क्रोनो. एनीबडी? मॉलिक्यूलर टैक्सोनॉमी और डीएनए बारकोडिंग में डिफरेंस वही मैम? क्वेश्चन वही है ना? Or what is the difference between this simple molecular detection or identification and molecular taxonomy? कोई बोलेगा?
So first time identification of something is like molecular.
ये आइडेंटिफिकेशन हो गया भाई. What is molecular taxonomy बोलिए? What is taxonomy? एक बंदा प्लीज. हाँ, अरेंजमेंट्स. डीएनए बारकोडिंग से वो सब एड्रेस कर सकते हो क्या? इफ यू आर हु सेड यस, क्या एड्रेस कर सकते हो डीएनए बारकोडिंग से?
So DNA barcoding molecularly we are characterizing one
पर्टिकुलर स्पीसीज.
plus. As you have told, like we are submitting the specimen motion numbers and morphological traits also. We are
That is just supporting your data for the sequence. See, the idea of molecular taxonomy per se is very very broad. So वहाँ पे you have to understand the interrelationship among the organisms, or species 1, species 2, species 3. If I want to understand a relationship between a Spodoptera and a Helicoarpa, my purpose should not be DNA barcoding. My purpose should be a molecular taxonomy where I will use other genes, multiple genes, okay, or genomics. आप लोग अभी ट्रांसक्रिप्टोम की बात कर रहे हैं, दोनों को ट्रांसक्रिप्टोम करूंगा डिफरेंट जगह से, मैं उसका एनालिसिस करूंगा. व्हाट इज द डिफरेंस, हाउ दे आर एसोसिएटेड, ओके, हाउ मेनी जीन्स दे आर वेरी क्लोज टू ईच अदर. That will give an insight between the how these two species are close or these species are far away from each other, how they have evolved. That is a broader aspect which is taxonomy or molecular systematics भी बोल सकते हो broadly. But DNA barcoding will not address that. DNA barcoding आपको बोलेगा मैं शशांक हूँ, बस मैं शशांक हूँ, that's it. शशांक लंबा है, मोटा है, DNA barcoding will not tell. For that you need molecular taxonomy. There you can add advantage with molecular taxonomy is it is amicable for morphological data also. Whatever data you generate for molecular taxonomy, आप morphological data भी उसको ऐड करके एनालिसिस कर सकते हैं. यहाँ पे उतना फ्रीडम नहीं है, ये एक या दो जीन से हम करते हैं, वेरी लिमिटेड. बट दिस इज इंपॉर्टेंट, दिस इज अ फर्स्ट स्टेप. एटलीस्ट कुछ भी, अभी 5% इंसेक्ट हमने डीएनए बारकोड किया है, ओके, सुपर पावर ऑफ द वर्ल्ड बनने जा रहे हैं, ठीक है. सो दैट्स द स्टेट एट विच आवर रिसर्च इज गोइंग ऑन. ये बहुत पुराना डाटा नहीं है, 23 का हमारा एनालिसिस है. उसके बाद भी अभी हम नया एनालिसिस कर रहे हैं. नथिंग हैज चेंज्ड, 0.1% एडिशन हुआ है बस. See what happens with that kind of mega analysis, it will give an idea, okay, where exactly we are lacking. Especially हम बहुत सीक्वेंस जनरेट किए हैं, लाखों सीक्वेंस जनरेट किए हैं, but we are concentrating on few species. White fly, we have more than 1.5 lakh sequences from India in bold, NCBI. For example, I am telling. So our focus has not been a holistic DNA barcoding always, it's species specific. That means principally we are not doing correctly. ये जो definition हम बोल रहे हैं ना उसको address नहीं कर रहे हैं. Okay, चलो बोर तो हो गया, 10 मिनट लेट हो गया. So this is barcode of life database system and यहाँ पे कोई भी आप लोग रजिस्टर कर सकते हैं. पहले जीमेल से भी रजिस्टर होता था, नाउ ओनली आई थिंक इंस्टीट्यूशनल ईमेल आईडी चाहिए. अभी ओनली एट द बिगिनिंग दे विल आस्क, वो ईमेल आईडी याद रखिए, बाद में कभी भी आप यूज़ कर सकते हैं. एंड द एडवांटेज हियर इज इन योर प्रोफाइल, इफ यू फॉर एग्जांपल नाउ यू आर अ स्टूडेंट, you are in IRI, tomorrow as a scientist you will join somewhere, you can change in the profile, that new update you can do, okay, because whenever you are submitting sequence from here to NCBI, those details are required, okay. So पूरा ये पोर्टल सीखने में 3 दिन चाहिए, ठीक है. Literally takes lot of time to learn, there is lot of things, but अभी मैं एक example, there are so many projects, not so many, there are 3 projects I have done in this portal, but I will just show you one project, okay. So project कैसे create किया जाएगा, उसमें what are the information we have uploaded, I will show you, okay. And how अभी तक हम बात कर रहे थे barcode, वो कहाँ पे आपको मिलता है, दिखता है, I will show you that. Before that I just want to tell you, ये जो barcode term आया ना, you know ATGC sequence, once you get a sequence it will be like 650 base pair or something, some length. So individual ATGC they will give it different colors, okay. You will see the barcode on any soap or something, it will be in black. Here it will be in 4 different colors or 3 different colors, depend on what you are using, okay. So it's very simple, it clear nahi hai na. So we have different projects here. I will just dwell into one of my project which is on Plucina of India. So before that, so this is a main console. So in main console it will show you how many projects you have, okay, and specimens, how many access to different records and how many barcodes you have generated. So if you want to start any new project, you can just create a project. हमारा थोड़ा नेट बहुत फास्ट है, you have to bear with us. So project code you can give, project title, for example like IRI, DNA barcoding of insects, pest diseases of IRI something. It will ask for primary markers. I told you, वो 30 steps बोला ना हमने, all steps are important because they will ask you all the details. So for insects what marker you are using, for pathogens what, जो first meeting में हमने, I just told you, first meeting all we sit and we will finalize what we are going to use, everything we can upload here, okay. So this is very important, which marker you are using. After that supporting markers. So what is supporting marker? So main markers, what I am talking, CO1, CO2, whatever we are using for insect, ITS. अभी मैम ने बोला फ्यूजेरियम के लिए टेफ यूज कर रहा है. So that will be the supporting marker at bold level, okay. Along with ITS they may be using, ITS तो यूज करना ही है उनको, है ना मैम, नहीं करना है, बोल्ड के लिए करना पड़ेगा. So supporting marker, then TEF will become supporting marker, okay. So there are so many options for you, of course. You can select whatever marker you require and they ask for campaigns. इंडिया में अभी ये ज़्यादा पॉपुलर नहीं है, this campaign system. In US, if there will be one mega funding agency, if they start some campaigns, they will give funding for like 100 institutes. So you have to claim that we are receiving funds from that campaign. That's why these slots they have given, okay. And of course containers are there. That means for example a mega project, DNA barcoding of like diversity in India, that's a mega project under which you want to work. So you can also add containers, okay. That's an advantage. For example here you can see aquatic fauna of Slovakia. There are so many mega projects are there, okay. and you can write, give a project description very short, and project access. So what is this project access? Many people, they don't want to share sequences publicly because अभी project खत्म ही नहीं हुआ है, शुरुआत कर रहे हैं. So you can restrict that to your own private. Okay. So if you give access here public, anybody can see, anybody can use your sequences. Okay. And also project manager, for example here in case of my, because default मेरा ही नाम आएगा, बाद में users assign कर सकते हैं. So my students, 3 students are working in same project, RAS are working. So you can assign them, they have to be registered in bold, उनका email डालते ही automatic assign हो जाएगा. So all of us उस project में काम कर सकते हैं इसी में. Okay, that's a good thing with this. That's it. This is very easy to start a project. And once you start a project, of course you start uploading the data. So there are so many tabs here, you can see in left. Okay, so I just want to go into this data which we have created. Barcode of what is that? Plucinia of India. It's a moth pest. Okay, pest group. So we have submitted so many sequences here. All sequences, sequence submissions. Yeah, there is add record here. 1 minute. Yeah. So you can directly go to projects here. You can view all the you can view all the projects here. These are all projects. Some projects we have just started, but there is no funding, so we have just stopped there itself. So, कौन सा है भाई? Okay. So I am going to okay. So this Indian Plucinia project is having around 74 specimens, sequences, okay, and all those sequences have specimen data. That's why it is showing 74 specimens and also 74 sequences, okay, and description of those, the description of the project. So what marker we have used? Cytochrome oxidase 1, VNT1, okay, and campaign general project, okay, those things we have added. There are about 23 bins, that means around 23 species we have, okay. So one of my student added the project, so I have also given access to her as a user. It will also show you the quality statistics of your sequences, what you have uploaded. आपने जो भी sequence upload किया है उसका quality statistics दिखेगा. And institute, for example, I am collaborating with 4 institutes. So 4 institute में 4 लोग 4 sequence किया है, सबका यहाँ पे contribution दिखेगा. That's an advantage. And also sequence age. This almost like 4 years back we have done. So it will show you when you have uploaded the data, okay. And specimen age. So whatever the data you have uploaded, collection of specimen details and all, it will give you. These data are very important for making a report. जो भी project करके project report तो बनना है ना. So what you will do? Generally manually करना पड़ता था, अभी यहाँ पे readymade है, just copy paste करो, project report बन गया आपका. ठीक है? That's an advantage with these tools. So if we go into each records, for example, this is a species. If I will show you the specimen page how it looks. Okay, this is a one of the species, Thesnoplusia aurichalcia. It's a common pest. Can you see this page? So this is a specimen, reference specimen we have used, and we generated data from this specimen, and all the data sample ID से लेके आप देख सकते हैं वहाँ पे sample ID, process ID, project ID and everything you can see there, and tissue description, from which tissue you have taken, legs, even male female we have written, okay, and taxonomy it will provide, bin number they have allotted, and also maximum distance in the bin, that means which is the nearest neighbor, इस species का nearest neighbor से कितना related है, you will also get that data, okay, and even also you can see from which location you have uploaded this specimen, you have collected this specimen, okay, from we have collected this from Uttarakhand. So, it will whatever data you have generated you have to upload in an Excel sheet, okay. So, everything will be shown here as a species data, okay, in a particular species. So, the next is sequence page. अभी तक just now we were always talking about barcode, okay. So, this is a barcode, illustrative barcode. So, I want to say that it is not only bold will give this illustrative barcode, there are so many tools nowadays, okay. You just type generate illustrative barcode for my sequence, there are so many tools, copy paste your sequence. For example, you have submitted to GenBank, but you want to generate a barcode, there are so many tools you can generate the barcode like this, okay. So, it will give nucleated in the sequence page, sequence view process, so many options are there here, bin URL is there and illustrative barcode and nucleated sequence what you have uploaded, and also it will give amino acid sequence here, okay. And GenBank accession, you can see metadata, this we have not shared with GenBank, otherwise even in GenBank accession number you will get here, okay. If you just submit it you will get here, and whatever amino acid just annotated there and showing as invertebrate mitochondria, okay. And what is that I can say, oh, sequence who has submitted the sequence, okay. So, all this data you will get in sequence records. It is very good way of arranging your complete project. So, this is a raw sequence you have, whatever the raw file you will get that you will upload, okay. So, it is called as ABA file. So, ABA file you will get along with your sequences, okay. Fine. So, here we have forward the details of primers have been provided here as LCO, HCO, and status you can see high quality sequence, so IRI. So, it's a very good platform, it takes some time to understand. So, I just want to show you about the bins, okay. I was just explaining you about bin. So, group of sequences. So, they have, I told you they have allotted a bin number to us. So, हमारा bin में हमारा एक ही sequence नहीं होता है, okay. There are so many specimens, you can see this, all these sequences generated from those specimens have been allotted this bin number, okay. So, you can also go to that bin, you can many people they have also analyzed bin diversity, many sequences they will download, they will also try to understand what is the diversity among the bins, okay. So, you can download if it is accessible. So, here you can see here, see the sequences in our bin are collected from samples are collected from different countries, you can see here contribution from different countries, you know this is a very like species is distributed in different places. So, you can see this, and also you can see the distribution distance distribution, pairwise distance analysis they have done. So, within bin diversity you can see here 0.2% within bin, the sequences whatever people have submitted, that is a within bin diversity. And the another one is NN plucine, nearby neighbor, nearest neighbor जो है इस species का, So diversity is around 3.8 to 4.2%. Okay. So you can consider why this value is important is, if you want to ID this particular species, you can't give a cutoff marks of cutoff of 5%. Generally you use 5% diversity variation. So 3.8 is enough, 3% you can use. So if you analyze, for example, 2 sequences from what is this, Thysanoplusia aurichalceae and other species, related species, if it is more than 3% variation, you can say that is a different species. Okay. So that is very important. You know how to calculate divergence, no? Class, okay, class होगा तो पता चलेगा. It's very simple. 100 nucleotides if you take, if there are 5 variations, 5% variation, it's very simple calculation. Okay. But there are so many statistical tools they will use. Fine. Okay. Few tools I will just show you. Not few, I will show you one, so that otherwise you can see here publication. You can also submit the project. You can get project summary and everything from here. Sorry. Okay. And you can download all these sequences and data spreadsheets from whatever project you have created. Sequence analysis is very important addition for BOLT, and this is unique. So अभी GenBank में ये सब options ही नहीं है. This is beauty with BOLT is there are so many tools you can use for your project. For example, I want to analyze a diversity between Thysanoplusia and Chrysodexis. I can select these 2 sequences and I can just analyze barcode gap analysis. I will just click barcode gap analysis, and there are so many criterias. I think bioinformatics class में आप ले सकते हैं. Now I am just taking default criteria. Okay. Apply default. Okay, I have to alignment. I have to select. I am just using muzzle. Last lo nam వచ్చేవా నువ్వు. Okay. Okay. So this is the analysis which shows difference between these 2 species, the sequences we have selected. Okay. I think it's not so clear for you. I'll just expand it. So this is maximum intra-specific versus nearest neighbor divergence. This is about 5.59%. It's visible now. Okay. So all these analyses are useful if you want to publish. Okay. This is individual per species. Anyway, individual per species हमने दोनों ही select किया है. But that is not in barcoding. You can't just sequence one specimen. You have to sequence multiple specimens, so that intra-specific variation also you should analyze. Okay. And next, this is a distance to nearest neighbor. Okay. So this is a distance to nearest neighbor 6%, because उसने average कर दिया यहाँ पे, because it's a percentage. Okay. So average करके 6% divergence it is showing. Fine. If you want to analyze multiple specimens, then this is a beautiful tool, that is barcode gap analysis, which is very important. There are so many analysis you can see here, even diversity measurements, accumulation curve, it's all important. And also you can generate, for example, you have uploaded like 50 species here, you can generate distribution maps for it. Okay. And you can generate it anyway, image library is there. Okay. You can also generate checklist. So 50 species का पूरा list आ जाएगा, उसका detail आ जाएगा एक ही इसमें. That's the beauty with this tool. Of course, there are so many like options here, but I इतना ही time है, 5 बज गया already. Any doubts you can ask me. So here they have provided a handbook of bold V4, okay, from scratch to final. There are 300, more than 300 page handbook. It's very well detailed handbook. If you are really if you want to work on DNA barcoding projects, okay, so you can really use that. Now at this point of time, the barcode of life database is not really bothered about single genes. Okay, the project क्या है वो? Project Life.
Life database.
हाँ, so they are now doing whole genome sequences for whatever organism on earth. It's a huge project of $500 billion. Okay, so it's a very huge project. By 2045 they want to complete. So that's the idea. You have a separate website for it because इतना handle नहीं कर पाएगा ये website. So they have a separate website for it, and because it's the pace of that project is like really stopped because of the COVID, because the main funders of these type of projects is European Union. Okay, so because of COVID they are also in trouble, but from next year I think it really kicks off again. So that's a great project to just follow. Any doubts? Please.
So what are the minimum number of sequences to generate that tree? Any minimum number is there?
Barcode. See, there is nothing like minimum maximum. It's up to you. So always I suggest to generate minimum 5 sequences. My thing is, आपके पास पैसा कितना है उसके बेसिस से करो. But 5 is a minimum you have to generate so that interspecific variation पता चलेगा. Another thing is, once you start work, एक ही सैंपल का एक ही PCR से 2 सीक्वेंस आप जनरेट करके देखो, machine errors बहुत easily पता चलेगा. So if you are starting with any private company, see most of the private companies Sanger sequence एक या 2 ही machine रहता है, ठीक है. If you are starting any work with them, the first thing you have to do is dummy samples 1-2 देना है उनको, एक ही PCR से 2 sequence करवाना है. That will tell you the machine error. Every machine is different. Okay, that machine error you will get to know the variations and all. Once you will get to know that, then you can really think about how many sequences you have to do. It depends on what project you are handling. Sometimes, especially in my case, I will tell, I am working on micromoths, where most of the times we are getting 1 or 2 specimen. So the specimen size is 10 mm. I can't even lose its scales also. You can't describe that species. इतना चौड़ा specimen है. Okay, and I have to use legs because 2 specimen है. I have to keep a holotype and a paratype for that. If I don't get a good quality DNA, even a short quality, now short DNA barcodes you can submit 300-400 base pair भी submit कर सकते हैं. Okay, at that time I can't really go with the number of sample. I don't have any option.
For new species you can reduce that.
हाँ, if you are very commonly occurring species and if you are working on that 1 or 2 species, try to get more samples. Diversity also you have to check. Location. I have seen many people, even at the early career, I have also done molecular diversity of Spodoptera littera using CO1. During that time, 1 gene was a big item for us. It's a huge thing. Literally, it doesn't give any signals with respect to variation. You should take 3-4 genes, minimum 5 genes, if you want to understand molecular diversity of a particular species in a location wise. इसीलिए मैं पूछा आप कोई पॉपुलेशन जेनेटिक्स कर रहा है क्या. आपको पॉपुलेशन जेनेटिक्स करना है, उसमें इफ़ यू आर यूजिंग CO1 इट्स नॉट अ गुड टूल. यस, 5 साल पहले यस, अभी आपको पेपर एक्सेप्ट भी नहीं होगा, ठीक है. इट्स ए वन ऑफ द जीन यू कैन यूज, बट उसके साथ मिनिमम 4-5 जीन्स चाहिए. ओके, अदरवाइज nowadays even mitogenomics is cheaper. You can directly go for mitogenomics. So you will get around 20-23 genes तो ऐसे ही मिल जाएगा. Okay, so there is always standardization is required, and also availability of your specimens you can decide. Any other questions?
What about eDNA and its status in insect pathology?
eDNA now people are using because it's required a lot of standardization methodologies. अभी microbes में standardize हुआ है, ठीक है. Insects में बहुत कम है अभी. So meta barcoding now we are standardizing. eDNA में अभी time है. Meta barcoding now we are just trying. We have now with what is that, Central Pollution Control Board, we are working on Namami Gange एक project से. Even there 90% of species we don't know even species identity. How you will name the barcode? आपको sequence तो आ गया है, but वो sequence का नाम क्या लिखना है हमें? So basic taxonomic data is important first. You can, we have wonderful tools now. Genomics has become so easy. I really now we are doing like 15,000 में आप एक genome generate कर सकते हो अभी. Okay. So it's a wonderful tool, but it's okay for pests. It's important to generate some data, but unknown species, especially for eDNA, using eDNA or metabarcoding, you should already have a library for it. You should know the number of species which are present in that ecosystem. Then only you can use how identifying those species. No, so we are in first stage. For insects, the first stage is first generate barcode libraries. Okay, later you can use those as a probes for metabarcoding.
We don't have secret. We can't think of e-secret. So first the secret only, then we can use for e-secret. So basic data in India is very less.
But it's a great tool. Nowadays eDNA, especially microbes में बहुत काम हो रहा है. Metabarcoding also, it's now NGS is giving so much of data. Okay. Yeah. So first thing is, it's a great opportunity. I say हमारे पास only 5% barcode हुआ है, means we are having a great opportunity. So दूसरा country में, why Europeans are coming to Asia for working in biodiversity? वहाँ पे छोटा country है, diversity कम है, they have done lot of work. So they are coming here so that there is lot of opportunity. So हमारा यहाँ पे, if this MSc PhD students, they can do small projects, some location, once they generate reference libraries, then that data will be useful. Next 4-5 साल में we can think about metabarcoding. Agroecosystem, it's very important. In future is, people will develop metabarcoding for identifying every agroecosystem में metabarcoding use होगा. That will be very common, because आपको 4-5 हज़ार में, if you can know the biodiversity of your field, people will spend. But you have to think about that, and at least in India we have to generate data or libraries for those aspects. Okay. Anything else? Okay, then 5 बज गया. And my suggestion is, because first you people are plant protection scientists, after that you are biotechnologists. Okay, पहले plant protection के angle में think करना है. As an entomologist, you think first as an entomology problem. एंटोमोलॉजी प्रॉब्लम को मॉलिक्यूलर टूल से कैसे एड्रेस कर रहे हो, पैथोलॉजी प्रॉब्लम को मॉलिक्यूलर टूल से कैसे एड्रेस कर रहे हो, दैट शुड बी योर थिंकिंग. व्हेन आई वाज इन पीएचडी, आई वाज आल्सो थिंकिंग वो CO1 बहुत बड़ा है, उस समय में बहुत बड़ा भी था. ठीक है, 2009 to 12 द DNA बारकोडिंग, एवरी पेपर इन एवरी जर्नल सिंपल DNA बारकोडिंग से आता था. 4-5 सीक्वेंस कर लो, दे यूज्ड टू एक्सेप्ट दैट पेपर. Okay, so now it is not the case. As an entomologist, you have to address your question with lot of data. Now you want to publish 2 to 5 impact में आपको publish करना है, तो data बहुत चाहिए. Only field data से काम नहीं चलेगा. Lot of molecular data you have to add. Okay, but when you are adding, be logical. So molecular tools, when you are applying now 10 days, you will learn superficially all the molecular tools. I am telling you, it is very superficial. आज qPCR कितने लोग एनालिसिस किया? सब लोग रखे ना, qPCR रख रहे थे. स्टैंडर्डाइजेशन सीखा, कैलकुलेशन सीखा. अभी कल सुबह में वो ही क्वेश्चन पूछूंगा, आप नहीं लिख पाएंगे, मैं लिख देता हूँ. सो द ट्रेनिंग इज जस्ट टू इंट्रोड्यूस टू दैट टॉपिक. वंस यू गो बैक, इफ़ यू वांट टू डू दैट वर्क, और एक बार मैनुअल डाउनलोड कीजिए QPCR कैसे कैलकुलेशन कैसे करना है, स्टैंडर्डाइजेशन कैसे करना है, एरर कैसे निकालना है. बाद में 10 बार फेल जब होंगे तब आपको एक्सपर्ट आएगा. फर्स्ट 10 बार सब कुछ अच्छा लगेगा आपको. ऑल रिजल्ट लुक्स वेरी गुड. ठीक है, एक बार पूरा होने के बाद आप यू विल ऑल्सो नो ओके दिस आर द मिस्टेक्स आई एम डूइंग. सो एनी ट्रेनिंग वी आर कंडक्टिंग, कोई भी ट्रेनिंग होता है, इट इज टू इंट्रोड्यूस द टॉपिक. ओके, सो ओपन माइंड से सब कुछ सीख के जाइए, बट आपको जो करना है आपको थीसिस के लिए वहां पे जाके रीस्टार्ट दैट पर्टिकुलर टूल फ्रॉम स्क्रैच. ये ए से जेड तक आपको पढ़ना पड़ेगा जाके. ठीक है, सो चलो ऑल द बेस्ट. टुमारो मॉर्निंग ऑल्सो आई हैव अ क्लास 1 hour. I'll see you. Okay, ma'am.
I think it was wonderful lecture, and you have exposed to another like domain which we are not thinking to submission of your sequence. Maybe next time when you work on diversity analysis or any kind of population genetics, you think of that you have another option to submit a sequence to board as well when you're submitting classes. Think in that way also. Even though you're not working in taxonomy, but you have the data to submit to the board as well. So that's also another part when you're working on the number of sequences. Along with the NCBI, you can also think of submitting to the board. Only taxonomists will not submit to the board. Also, people are not working in taxonomical area. Also think about something like this.
Okay, thank you. कल मॉर्निंग 10 o'clock I have another topic. It is completely different. इस पर कितने लोग जो वेडिंग में काम कर रहे हैं? इस पर का क्लास हुआ कोई? इंट्रोडक्शन तो हो गया होगा, आधा काम हो गया. So one application, so we will discuss tomorrow. इतना CRISPR का चल रहा है ना, एक छोटा